As you saw in lecture 4, the sequence of nucleotide bases is really important for determining the differences between living organisms. And those differences can be between members of different species, so like the previous example, the difference between cats and crabs and humans, or subtypes of the same species. So what makes, a Chihuahua different from a Great Dane? We're going to talk a lot more about dogs a little bit later in the next lecture. Or what makes individuals of the same species different from each other. So even between two humans, the differences between individuals is down to a difference in the sequence of A's, C's, T's, and G's in the DNA of those people. And if we look over here on the right, these are little snippets of a gene that codes for insulin. So this is a sequence of DNA that codes for insulin. You might know insulin as the compound that is responsible for blood sugar, and it sends a signal to cells, tells them to take up glucose from the bloodstream into the cell so they can use it for cellular respiration. And in most people, the gene for insulin is going to have the exact same code because everybody uses it for the exact same purpose, and it's going to be the exact same shape in all of those people, and it's going to have the exact same intermolecular interactions in all of those people. There might be a little bit of variation within the population, like maybe, one nucleotide difference here or there. And so we can see here between human A and human B, there is a single section of the gene that codes for insulin that has a mutation in it, or that has a difference within it. Whereas if we looked at the difference between the insulin gene in a chicken versus in a person, there's a whole lot more differences because chickens and people are genetically divergent. Two people are more similar to each other in terms of their DNA than a person and a chicken are. Chickens still have insulin for the same reason that we do, but they have a slightly different code for that insulin, and that insulin would be a slightly different shape than our insulin and would have slightly different intermolecular interactions than our insulin does. All right, and I've already used this term genome a couple times, so I want to make sure we're all on the same page about it. But basically, the genome is just all of the genetic information stored inside the nucleus of all the cells in your body. And humans have two sets of chromosomes. We have 23 chromosomes times 2. And if we added up all of the nucleotides in all of our chromosomes, we have 3 billion base pairs. 3 billion base pairs in one of the sets of 23. And so from our mom's egg and from our dad's sperm, inside each of these cells, there were 23 chromosomes. And when these came together to create the zygote that became us, to be the fertilized egg that became us, now there are 46 chromosomes in here. There were 3 billion base pairs in there, 3 billion base pairs in there, and so 6 billion in here. Just a monumental amount of information. So many A's, C's, T's, and G's that are writing traits that we have in our bodies. I'm going to put this video as an optional link after this lecture video. It's just a kind of neat TED Talk. A guy called Riccardo Sabatini, he talks about how mothers, when they're gestating a embryo in utero, they're basically acting like 3D printers. And so the genetic information inside of the zygote, inside of the fertilized egg, is building a human based on these sequence of A's, C's, T's, and G's. But in terms of, practicality and what we can use genetics for, beyond just saying like, oh, that person's hair is brown because they have this gene and my hair is blonde because I have this gene, we can use genetics to do a whole lot of things in terms of culture and science, such as paternity testing. So maybe you've heard of paternity testing and you know that you could get a DNA sample from a mother, a child, and potential fathers, and then match the DNA from the parents to the offspring. You can do maternity testing just like paternity testing. So if you didn't know who the mother of a child was, you could compare the DNA of mom to the kids the same way you compare the DNA of dad to the kids. And the way that this works is because we all have slightly different sequences of A's, C's, T's, and G's, we can run what's called a DNA fingerprinting test where we get a unique banding pattern depending on our own DNA. So if we look at mom here, mom, because of her DNA, is always going to have this very specific banding pattern right here. If you extracted the DNA from mom 20 times, all 20 times when you ran that DNA out on a gel, you would see this exact banding pattern. And then if we look at dad in this family, he's also going to have a distinct banding pattern. So every single time you run out his DNA in a DNA fingerprinting test, his banding pattern is going to look like that. If this woman and this man had a child together, because the child is the result of an egg from mom and a sperm from dad, the child is only going to be able to have a combination of bands from mom and bands from dad. So if we look at daughter number one is the result of a egg from this woman combining with a sperm from this man. If we look at daughter number two did come from an egg from mom. You can see that this band matches up with this band matches up with that one, this one with that one. But daughter number two did not come from a sperm from this father here. There was another man, not pictured here, who would have donated the genetic information that you see in red there. Son number one is the genetic result of these two parents, just like daughter number one. You can see the yellow bands match up with dad's sperm, and the blue bands match up with mom's egg. And then son number two, he doesn't match either parents in his banding pattern, so he would be the product probably of adoption in this scenario. This exact same science applies to DNA fingerprinting in terms of forensics. And so we already kind of talked a little bit about, when we talked about PCR, like if you're at a crime scene and you collect DNA from the crime scene, you need to amplify it. But then once you've amplified it, if you want to match it to suspects, you use the same science to do that. You do a DNA fingerprinting assessment. So you would take, say, a blood sample from the crime scene, and you would fingerprint it, and you get a very specific banding pattern because the bands are always going to be the same for each individual person. Just like mom's bands always look like this, if the blood at the crime scene came from this woman, the banding pattern would look exactly like this, regardless of whether you were testing her for maternity or whether you were testing her presence at a crime scene. So if you take blood from a crime scene and you do a DNA fingerprint, and then you take blood from 3 suspects or cheek cells from 3 suspects, if you get DNA from suspects, you can run out their DNA fingerprint and then look for a match. And so in this case, can you spot just by eye who was at the crime scene in this scenario? Yeah, suspect 2. So if you compare these two together, they are identical. And so in DNA fingerprinting in forensics, you're looking for an exact match. You're looking for DNA found at a crime scene and comparing it against DNA of potential people that could have been there. When you're doing it with paternity testing, you're looking for a half match. You're looking at DNA from a child, and knowing that half their DNA came from mom and half came from dad, you can try to match up those bands with either or both parents. We can also use our genomes and genetic information to assess ancestry. Maybe you've done 23andMe or Ancestry.com, something like that. What those sites are doing is looking at portions of your genome, looking at portions of those 2 sets of 3 billion base pairs, and looking at regions that are known to vary geographically. And then they compare your DNA to a reference population in different parts of the world. So a reference population from Western Europe, or a reference population from East Asia, or South Africa, or anywhere else on Earth, Australia. And then they look at how similar you are to those reference populations. And so when they say, if you get your results and they say like, oh, you have a 27% ancestry in Western Europe, they're not saying that you are 27% European. They're saying that you are 27% similar to people in the reference population in that part of the world. And if you've done this, a couple years ago, reference populations change over time. And as more information gets added to the database, your numbers can actually change over time. So if you did this, 5 years ago and you haven't looked since then, go back and have a look now, because it's very likely that you might have come up, 30% South African 5 years ago, and now you might be 40%, or now you might be 20%, depending on how the reference population has changed through time. So genomes are not, in terms of ancestry, are not as steadfast a science as forensics or paternity testing is. This is more of a hand-wavy kind of relationship that is being used here by these companies like 23andMe and Ancestry.com. The other way that we can use genetic information to classify organisms is through taxonomy. And I like to say that 1107 is how all life is the same, the course you're taking right now. Like, all living things have the 4 macromolecules inside of them. They've all got DNA, all that jazz. And when you take 1108, if you take 1108 after this, 1108 is about how all life differs and the diversity of life on Earth. And it journeys through DNA relatedness between individual organisms. And all of that is down to just looking at the difference in A's, C's, T's, and G's in animals versus, let's see what else is on here, plants or archaea or bacteria. All of these organisms have DNA that's just a sequence of A's, C's, T's, and G's. But the number of base pairs that they have and the sequences of those base pairs is going to be a big difference between like E. coli growing in chicken on your counter versus you, a human being. If we want to think about how DNA is stored in living cells, we will just quickly kind of outline what's going on in prokaryotes and eukaryotes, and then we will move on to the next topic in the next video. So prokaryotes, you'll remember from Unit 1, are cells that do not have a nucleus or any organelles. And prokaryotes are going to be things like bacteria. And so in bacteria, the DNA is not stored in a nucleus, it's just stored in the sort of middle of the cell within the cytoplasm. And DNA in bacteria is circular. So they've got one chromosome. Remember, we had 23 chromosomes, 2 sets of them. Bacteria just have one. And so it's this big kind of circle helix. The DNA is still a helical shape, but it's connected at the ends. So you can kind of think like our DNA is twist ties, you know, they're linear, whereas bacterial DNA is more like a rubber band. It connects back on itself. And all bacteria are going to have like one main chromosome, and then they might also have tinier plasmids, which are like little extra bits of information they've picked up through time. And the genomes of bacteria are pretty small compared to eukaryotes. The genomes of prokaryotes are pretty small compared to eukaryotes. You don't need to recognize any of these names here. These are just different species of archaea bacteria, different species of prokaryotes. But you do want to take a second just to look at this x-axis down here. So genome size in MBP. Million base pairs. So our genome was 3 billion base pairs. The largest bacterial genome is only about 10 million base pairs. So much, much, much smaller than us. Way less genetic information than we hold. And it makes sense because we are a much more varied individual than a single bacterial cell is. But what this means for bacteria, because they're tiny and because they don't have that much DNA, they can copy their DNA way faster than we can, and they can divide way faster than we can. So it takes a bacterial cell about 20 minutes to divide and copy its DNA. It can take our cells weeks to do the same thing. And then if we think about what's happening in our cells or what's happening in eukaryotic cells in general, remember I said that our DNA is more like a twist tie than a rubber band. Our DNA is linear, and our DNA is arranged into these 2 sets of 23 chromosomes. So at the smallest portion, we've still got a double helix. And that double helix is wrapped around these proteins called histones. And then these histones kind of wrap around themselves like a, old-timey phone cord. And then that kind of wraps around itself and bundles into this chromosome structure that you see down here. And we're going to talk a lot more about chromosomes specifically a little bit later in this lecture. But when we talk about our cells having 23 sets of these, so 46 total chromosomes, each of those chromosomes is a big bundled mess of lots and lots and lots of DNA here that we laid out in lecture 4. So our genetic code is super, super, super tiny in this very small DNA, and then it's all wrapped up into these larger chromosomal structures which are stored in the nuclei of our cells. If you were to take all the DNA in one of our cells, in one human cell, and spread it out, take all 2 sets of 23 chromosomes and unwind all of these bits and just spread out DNA in this orientation end to end, you would end up with about 6 feet of DNA in every single one of your cells. So we have this sort of masterful folding mechanism that allows us to store tons of information in every single tiny little cell in our body. All right, so there'll be a question after this video, a couple of optional videos to kind of help you nail down some of those ideas, and then we will move on.